Hi, my name is Alfredo Huerta, Professor Emeritus of Biology at Miami University in Oxford, Ohio. Today's lecture is titled Glycolysis, Part 1. Glycolysis is the process by which glucose is broken down in cells in a series of biochemical steps. In this lecture, I will provide you with the necessary foundation for you to develop a basic understanding of glycolysis. A second lecture, which I have titled Glycolysis Part 2, will cover each of the reactions of this biochemical pathway in more detail. Glycolysis comes from glyco, the Greek glyco, which means sweet or sugar, and lysis, which means to break apart. Glycolysis is a biochemical pathway that breaks down the 6-carbon sugar called glucose as a source of carbon and energy. It occurs in almost every type of living organism that we know of. Let me explain that a little better. Biologists used to classify living organisms into kingdoms such as the animal kingdom, the plant kingdom, the fungi kingdom, etc. However, with modern advances in fields such as genetics, developmental morphology, cell biology and evolution, and computers, coupled with better fossil evidence, biologists now divide all organisms into what we call the 3 domains of life. Those 3 domains of life include the bacteria, the archaea, and the eukaryota. The archaea include types of organisms, that would be the one here in the middle. The archaea include types of microorganisms that are able to live in extreme environments, such as deep sea and hydrothermal vents. Now, the Earth is about 4.5 billion years old, and the first living cells are thought to have evolved just over 3.7 billion years ago. And that would be the common ancestor here on the bottom, giving rise to all life on Earth. As shown in this diagram, evidence indicates that all organisms on Earth evolved from those primitive cells, or some common ancestor. In another lecture, I will describe the major characteristics of the organisms in each of the 3 domains of life. Now, I should point out here that a few types of microorganisms in the bacteria and the archaea domains of life do not perform glucose breakdown as described in this lecture. Okay, that's just a few types, not all of them. Those organisms can extract energy from glucose using alternative reactions, okay, different reactions from the ones I will describe here, or they extract energy from completely different molecules such as hydrocarbons. So you can see where animals and plants are located. Animals, fungi, and plants are organized into the eukaryota. As I said before, I will talk about the details of how the organisms are divided into these 3 domains of life in a future lecture. So let's get started with glycolysis. Glycolysis begins with glucose as the very first substrate of this biochemical pathway. During this pathway, each molecule of glucose, composed of 6 carbons, okay, that's a 6-carbon molecule, is broken down to 2 molecules of pyruvate, each composed of 3 carbons. So you get 2 of the 3-carbon pyruvates. Now, during the biochemical reactions of glycolysis, 2 molecules of NADH are synthesized. NADH is an electron carrier that can receive electrons as it is reduced, and then donate electrons as it reduces other molecules. In addition, 2 molecules of ATP are synthesized during glycolysis. ATP is a molecule that provides phosphate or energy to many endergonic or energy requiring biochemical reactions, allowing those reactions to proceed. Without ATP, those reactions would not occur, and a cell could not remain alive. Also notice that no oxygen is consumed during the process of glycolysis. As I mentioned earlier, glycolysis begins with glucose and ends with pyruvate, but in no place during the process of glycolysis is oxygen consumed. I will cover the structure and mechanisms of synthesis of NADH and ATP in a separate lecture. Now, the two pyruvate molecules, which are the end products of glycolysis, that would be these two down here, these two pyruvates, they can't accumulate indefinitely in a cell. The pyruvate has to go somewhere. Now, how the pyruvate is eliminated or processed depends not only on the type of cell, but on the conditions under which this process occurs, such as the presence or absence of oxygen. That said, in this lecture I will cover only the reactions starting with glucose and ending with pyruvate. In a separate lecture I'll discuss the fate of pyruvate as it is metabolized into other products, but I'll provide a very brief overview here. So just to refresh your memory here about what I said, NADH is an electron carrier that can receive or donate electrons, and ATP is a phosphate and energy carrier. So remember those. So let's start with glycolysis. And I have circled or formed a square around glycolysis here so we can focus on that. Now, glycolysis is only part of what's known as central metabolism in cells, which includes the citric acid cycle, with those things that appear, I'll talk about those in a second, and oxidative phosphorylation. After glycolysis, the breakdown of pyruvate under aerobic conditions, that means under conditions of having oxygen available, what happens is a series of reactions called the citric acid cycle. So this citric acid cycle, also called the Krebs cycle or the tricarboxylic acid cycle, and I'll explain why those alternative names occur in my lecture covering citric acid cycle. Now, these processes occur in the presence of oxygen, as I mentioned. No oxygen is consumed in the citric acid cycle, so in this cycle, no oxygen is consumed, but you'll see that oxygen is consumed later during oxidative phosphorylation, which works in conjunction with the citric acid cycle. Now, during entry into and as part of the citric acid cycle, all of the 3 carbons of the 3-carbon pyruvate are lost as free CO2. So here is the 3-carbon pyruvate, and when this 3-carbon pyruvate enters into the citric acid cycle, CO2 is lost in the process at the beginning and then later on during the cycle, during the citric acid cycle. During this cycle, for every glucose that is broken down, okay, beginning with this glucose up here, for every glucose that is broken down, 8 NADH molecules are synthesized within this citric acid cycle, or at least as part of it. 2 when pyruvate enters into the citric acid cycle, and then 6 during the citric acid cycle. So we have a total of 8 NADH molecules being synthesized from every glucose. In addition, 2 molecules of FADH, 2, which is another electron carrier molecule similar to an NADH, are synthesized. Also, 2 more molecules of ATP or GTP are synthesized. So here on the bottom left we have ATP or GTP. We have 2 of those being synthesized for every glucose. And turns out that GTP is similar but not identical to ATP in structure and function, but it works to power different reactions that ATP doesn't. So GTP itself functions in specific reactions that require GTP. Now the last part of central metabolism is called oxidative phosphorylation, which we have here on the bottom. This last step requires the input of NADH and FADH2 synthesized earlier. In other words, this NADH, this NADH, this FADH2, and this NADH, all of those enter into oxidative phosphorylation, donating electrons. And the result is that because of oxidative phosphorylation, during which oxygen is consumed, 34 ATPs are synthesized. Again, I'll cover oxidative phosphorylation in a future lecture, just like I'll cover citric acid cycle in its own lecture. Now, central metabolism is called central metabolism because it plays such a central role in energy and what is called carbon skeleton processing in cells. A carbon skeleton is generally thought of as any molecule of central metabolism that can be used by other pathways as a source or as an end product. Many important metabolic pathways involving the synthesis and breakdown of amino acids and proteins, lipids and fats, carbohydrates, and nucleic acids interface with central metabolism. This happens because some of the intermediate substrates or products of glycolysis and the citric acid cycle can be used as carbon skeletons, as I mentioned earlier, by those secondary pathways. So all of these different pathways and others not mentioned here connect to central metabolism in one way or another at some point during glycolysis or the citric acid cycle. So let's get down to the business of focusing on the process of glycolysis. First, where in a cell does glycolysis occur? Glycolysis occurs in what we call the cytoplasm of the cell, a region of the cell filled with an aqueous solution containing many types of organelles. Now, organelles are membrane-bound bodies which perform specific functions in a cell. Now, examples of organelles are the nucleus, mitochondria, Golgi bodies, chloroplasts, etc., depending on the type of cell that exists. So glycolysis occurs in the cytoplasm of a cell, as I mentioned, and the cytoplasm is always surrounded by a selectively permeable membrane called the plasma membrane in all living cells. But in plants, you will hear plant biologists sometimes call it the plasmalemma. Now, for illustration and orientation, this is the organelle called the nucleus, which, as described, is surrounded by its own membrane called the nuclear membrane. Certain types of animal cells, cancer cells, plant and algal cells often contain more than one nucleus, but I'm drawing only one nucleus in this cell. The nucleus contains most of the genetic material in a cell in the form of DNA organized into chromosomes. Another type of organelle in the cytoplasm is the mitochondrion, the plural being mitochondria. Mitochondria contain the reactions involved in the citric acid cycle and also oxidative phosphorylation, as mentioned earlier. Mitochondria also contain their own DNA separate from the nuclear DNA. It's called the mitochondrial DNA. Plants contain DNA in a third type of organelle called the chloroplast, the site of photosynthesis. That's something that I'll cover in future lectures. Now, I have highlighted the cytoplasm in a tan color to indicate that it is separate from the organelles that are contained in it. In other words, the contents of those organelles is kept separate from the cytoplasm by the membranes that surround each organelle. So here is the membrane that surrounds the mitochondria, and here's the membrane that surrounds the nucleus. And so the cytoplasm is separate from the interior of those organelles because of the presence of that membrane or those membranes. You can see then that the cytoplasm is the actual site of the reactions, the biochemical reactions of glycolysis. As stated earlier, it is these reactions that I will focus on during the rest of this lecture. So, how glucose is converted into 2 molecules of pyruvate, these reactions are what I will cover here. Before I get going on how glucose is broken down in the cytoplasm of a cell, I should first explain where the glucose comes from. It just doesn't appear in the cell. It has to come from somewhere. First, plants make their own glucose from inorganic products. So plants are photosynthetic, at least most of them. There are such things as albino plants or something similar to an albino plant, plants that are not photosynthetic and they are parasitic. They can extract their organic molecules from other plants. But most plants are photosynthetic. Plants make their own food by synthesizing organic molecules from inorganic molecules such as CO2 and H2O, and also from minerals. For this reason, plants are known as autotrophs. In other words, autotrophs means that plants feed themselves. Green tissues of photosynthetic plants absorb solar energy, which drives the process of photosynthesis in chloroplasts. So CO2 plus H2O plus the minerals absorbed, powered by absorption of energy from the sun, results in the synthesis of 3 carbon sugars. Now glucose is synthesized later. Okay, and I highlighted the word later here because some people get confused about when or where glucose is synthesized. Glucose is synthesized later or after those 3 carbon sugars are synthesized during specific reactions that are not part of the biochemical processes of photosynthesis. So outside of the process of photosynthesis, glucose is synthesized from those 3 carbon sugars. And that can happen either inside the chloroplast or in the cytoplasm. Now, after synthesizing glucose, plants can store it in several forms, but the 2 major storage forms are starch and sucrose. So, starch is a polymer formed by many glucose molecules linked together. So you have one glucose molecule linked to another glucose molecule, linked to another glucose molecule, and then to another, etc., 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 forming these chains that you see in the representation that I have here, this starch molecule that I drew. Kind of like a chain of beads into long branched chains. Now 80% of the calories, it turns out, consumed by humans around the world come from starchy foods, such as rice, wheat, maize, potatoes, and cassava. The other common storage form of glucose in plants is sucrose. Sucrose is common table sugar, which is synthesized in large amounts in plants like sugarcane and sugar beets. Sucrose is a molecule formed by a covalent bond between 2 different monosaccharides, and those monosaccharides are glucose and fructose. Now the word fructose comes from fruit, because in fact many fruit are high in this monosaccharide, or this sugar called fructose. And when glucose and fructose come together by this oxygen, with the covalent bonds linking the glucose and the fructose, that's called a dimer. And a dimer is a molecule formed by 2 similar but not necessarily identical subparts. So in this case we have glucose, which is a monosaccharide, and fructose, which is a monosaccharide. They're not identical, but yet they form a dimer, and this is a dimer of two monosaccharides. However, as I said earlier, many fruits, for example apples, grapes, cherries, etc., contain a significant amount of free fructose, and they don't have large amounts of sucrose, so it depends on the type of fruit. The breakdown of starch, sucrose, and many less common sugars releases individual sugar molecules that can be broken down in cells throughout the plant, providing carbon sources and energy. So in other words, when this sucrose breaks down, it will release free glucose and free fructose. And then those sugars become available in the cells that consume them. However, the question now is, where does glucose come from in animals? Where do animals get their glucose from? Animals are not photosynthetic. So for an animal to live, it must consume organic molecules previously contained in other organisms, such as other animals, plants, or other photosynthetic organisms such as algae and cyanobacteria. Because of this, animals are known as heterotrophs. In other words, they feed themselves from other things previously existing. They don't feed themselves with things that they synthesize themselves, like plants, which are autotrophs, as I mentioned earlier. So, consumed starch, fructose, sucrose, and many other types of sugars, which are all carbohydrates, and there are many other types of sugars, can be used as carbon sources and as fuel. Now, when molecules other than carbohydrates, such as proteins and fats, are consumed and broken down, the resulting products of the breakdown of those proteins and fats can enter into pathways of central metabolism at specific biochemical steps that are part of either glycolysis or the citric acid cycle, as I mentioned earlier. Now, sugar consumed as fructose, for example, when you eat fruit, or fructose that is released from the breakdown of sucrose in the small intestine, is mostly transported to the liver, where up to half of it is converted to glucose. So here we have fructose being converted to glucose in the liver. Now, the free glucose released in the liver can then be used to form glycogen. Glycogen is a polymer form of glucose storage, very similar to starch. So glycogen in animals is extremely similar to starch in plants. However, glycogen is more highly branched than starch, and so I drew this molecule with many more branches. You can see all the little branches here, much more branched than starch. And in addition, the covalent glucose-glucose bonds of glycogen are different from those of starch. I won't cover those details here, but they are different. Interestingly, glycogen is stored in liver and muscle cells, and when needed, that glycogen is broken down to individual glucose molecules. So the glycogen is broken down when needed, and the individual glucose molecules are transported to target cells via the bloodstream. Now, what happens at target cells? Well, at target cells, the glucose is broken down via glycolysis, and that provides energy and carbon sources to those target cells. It's also interesting to note that both animals and plants can also synthesize glucose by a series of reactions called gluconeogenesis, but I'm not going to cover gluconeogenesis at this time. I will cover it in a future lecture. So reviewing on the bottom here, glycogen, which is a storage form of glucose in liver or muscle cells, is broken down to free glucose, and that glucose is transported via the bloodstream to target cells where it can be broken down by glycolysis, providing energy and, as I mentioned earlier, carbon skeletons to those cells. Another interesting point is that the brain basically lives on glucose. The brain consumes about 50% of all the available energy in the body that is derived from glucose. So I have a little drawing of a brain here, and you can see glucose coming into the brain, and because of that, the brain can think. So let's get back to talking about the actual process of glycolysis. Glucose is formed as a molecule with 6 carbons, and as I had mentioned earlier in one of my previous lectures, those carbons are numbered 1, 2, 3, 4, 5, and 6. And energy is required in order to synthesize the covalent bonds that hold the atoms of the glucose molecule together. In other words, these covalent bonds, all of these covalent bonds, take energy to form. And when glucose breaks down, energy is released in the form of heat, or it can be conserved in the bonds of other molecules. And lastly, that complete glucose breakdown to CO2 and H2O releases a total of 2,840 kilojoules per mole of glucose, which is the same as 678.76 kilocalories per mole. Now, I covered this information in one of my previous lectures, specifically titled, How Much Energy Is There in 1 Mole of Glucose? So if you like, go ahead and watch that lecture and you'll get more details about this. So again, getting back to the actual process of glycolysis, glycolysis occurs in the cytoplasm of a cell in 10 enzyme-catalyzed steps, and each one of those steps requires a different enzyme. Okay, here we have glucose, which is a 6-carbon molecule, and in many steps, 10 of them, eventually producing 2 pyruvates on the bottom here, each with 3 carbons. That results in the net synthesis of 2 molecules of NADH and 2 molecules of ATP. So here we have glycolysis, 10 steps. Now let me refresh your memory that we're only talking about this part of central metabolism, right here, where glucose is broken down to 2 pyruvates with the synthesis of 2 NADH for glucose and 2 ATPs for glucose. That's all we're talking about here. So following then is a simplified version of glycolysis. A more complete version of the 10 steps of glycolysis will be presented in the lecture titled Glycolysis, Part 2. So let's begin. Turns out that biochemists divide glycolysis into 2 separate phases. The first phase of glycolysis is composed of reactions 1 through 5. So here on the left we have phase 1 with steps 1 through 5. The green numbers to the left of each substrate or product, as I have drawn, indicate how many molecules are involved. So here we're starting with 1 glucose, which makes 1 glucose 6-phosphate, which makes 1 fructose 6-phosphate, which makes 1 fructose 1,6-bisphosphate, which makes 1 glyceraldehyde 3-phosphate, and 1 dihydroxyacetone phosphate, and between those two it forms 2 glyceraldehyde 3-phosphates. Each of the steps of glycolysis is numbered 1 through 10 with a red number. Phase 1 is generally called the energy investment phase, since ATP is required to move these reactions forward. So looking at these red numbers, that's step number 1, step number 2, 3, 4, and 5. So let's look at each one of those steps. In step 1, that would be this one here on the top, ATP, this molecule here, which stands for adenosine triphosphate, and I will cover the details of each of these reactions and the enzymes involved and the structure of all the molecules and stuff like that in detail in the lecture that I'm titling Glycolysis Part 2. So in step 1, ATP donates a phosphate atom to the original glucose substrate, which is a 6-carbon sugar. So here we have glucose, which is a 6-carbon sugar, and the ATP, which we have right here, donates a phosphorus to the glucose, and the result is the phosphorylation, or the addition of phosphate to the glucose molecule. So the glucose molecule becomes phosphorylated, and the result of that is glucose 6-phosphate. Now the incoming phosphate is attached to carbon 6 of the glucose molecule, and that is denoted in the name of the molecule, glucose 6-phosphate. You can usually tell which carbon the phosphate is on by the number in the name of the molecule. So if it's called glucose 6-phosphate, right away you can generally conclude that the phosphate is on carbon 6. Further down, between reaction 3 and reaction 4, we have fructose 1,6-bisphosphate. That means that there are 2 phosphates, that's called bisphosphate, and those phosphates are attached to carbons 1 and 6. So, getting back to where we were, the ATP that lost the phosphate is converted from adenosine triphosphate, or ATP, to adenosine diphosphate, or ADP, because at first it had 3 phosphates, then it has 2 phosphates. Now in step 2, that would be this step right here, the glucose 6-phosphate is converted to fructose 6-phosphate. Okay, so in this step right here, glucose 6-phosphate to fructose 6-phosphate. And fructose 6-phosphate is also a 6-carbon sugar. This step consists of a reorganization of the atoms of the phosphorylated glucose molecule into a phosphorylated fructose molecule. That's all that happens. Moving on to step 3, right here. In step 3, the fructose 6-phosphate is phosphorylated by another ATP molecule in a process similar to step 1 as described above. So again, we have an ATP donating a phosphate to the fructose 6-phosphate, resulting in the synthesis of ADP, and fructose 6-phosphate being phosphorylated to form fructose 1,6-bisphosphate. Again, as I said earlier, the phosphates are on carbons 1 and carbon 6 of the fructose molecule. Now steps 1 and step 3 involve the investment of energy as phosphorus in the form of ATP, and this is why phase 1 is called the investment phase, or the energy investment phase. Now in step 4, the fructose 1,6-bisphosphate is broken down to 2 separate 3-carbon molecules. So reaction 4, or step 4, this fructose 1,6-bisphosphate is split. And it's split into the 2 molecules, one of them being called glyceraldehyde 3-phosphate, often abbreviated as G3P, but you'll find it abbreviated as GAP, and GAP stands for glyceraldehyde phosphate. But depends on which book you look into, you'll find it, again, either G3P or GAP. And the other molecule that it splits into is dihydroxyacetone phosphate, abbreviated as DHAP. So we get 1 G3P and 1 DHAP. Now it turns out that only the glyceraldehyde 3-phosphate can be processed further in glycolysis. The dihydroxyacetone phosphate cannot be processed. So this molecule here, DHAP, cannot be processed further directly in glycolysis. So what happens is that step 5, this one down here, converts the DHAP to glyceraldehyde 3-phosphate in this direction. So at the end of this step, the result is 2 molecules of glyceraldehyde 3-phosphate. One of them coming directly here, and the other one coming directly here. So we end up with 2 molecules of glyceraldehyde 3-phosphate. So I just finished describing phase 1 of glycolysis. Steps 1 through 5, in which 1 molecule of glucose, and here's the 1 molecule of glucose, is broken down to 2 molecules of glyceraldehyde 3-phosphate. And during those 5 steps, energy was invested as 2 ATP molecules, which were used to phosphorylate substrates at steps 1 and 3. So there's the ATP at step 1 and the ATP at step 3. And we can now move to phase 2 of glycolysis. This is generally called the energy recovery phase because energy is generated in the form of 4 ATPs and 2 NADHs per glucose. Now let's see how that happens. Note that since this phase begins with 2 molecules of glyceraldehyde 3-phosphate, and here they are, 2 molecules on the top of glyceraldehyde 3-phosphate, each step, 6 through 10, okay, here's 6, 7, 8, 9, and 10, happens twice, once for each of the glyceraldehyde 3-phosphate molecules, resulting from the breakdown of glucose in phase 1. So let's start with step 6 of glycolysis, right here on the top. During this step, the end product of phase 1, the glyceraldehyde 3-phosphate, that would be this one right here on the top. This is the end product of phase 1. During step 6, the glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. So you can see that glyceraldehyde 3-phosphate has 1 phosphate on carbon 3, but the following product has 2 phosphates on it, 1 on carbon 1 and 1 on carbon 3. So it's called 1,3-bisphosphoglycerate. So has 2 phosphate atoms attached to it. The phosphate atom in this case does not come from ATP. So if you look over here on the right, there is no ATP. But instead, the phosphate comes from free phosphate ions floating around in the solution. The PI, right here, is noted here on the top right, stands for inorganic phosphate, and its structure is chemically named orthophosphate. So whenever you see PI, think of the name orthophosphate. That's what it's called. It's just inorganic phosphate. It's actually phosphorus attached to several oxygens. Now during this phosphorylation reaction in step 6, electrons from glyceraldehyde 3-phosphate are transferred to a molecule called nicotinamide adenine dinucleotide in the oxidized form. So that's abbreviated as NAD+. The plus indicates that it is in the oxidized form. So this is an example of oxidation reduction. During the process of electron transfer from glyceraldehyde 3-phosphate to NAD+, the glyceraldehyde 3-phosphate loses 2 electrons and is oxidized as a result. At the same time, the NAD+ gains those 2 electrons, so we say that the NAD+ is reduced or becomes reduced to NADH as a result, so that NAD+ is reduced to NADH. Don't pay attention to this other hydrogen at this moment. Just think about the fact that the NAD+ is reduced to NADH. And I'll explain this in my future lecture on glycolysis part 2. Remember that this reaction happens twice. There are 2 here and 2 here. Once for each glyceraldehyde 3-phosphate. So in fact, 2 NADH molecules are produced in step 6 for each glucose broken down. this glyceraldehyde 3-phosphate is energetic enough and reduced enough such that when it loses electrons to the NAD, okay, there it is losing electrons to the NAD, 2 of them, there's sufficient energy released in this reaction to take an inorganic phosphate and attach it to the molecule, resulting in the 1,3-bisphosphoglycerate. So, moving on, in step 7 of glycolysis, the 1,3-bisphosphoglycerate, this one here, loses one of the phosphate atoms and becomes 3-phosphoglycerate, abbreviated as 3-PGA. So the 1,3 becomes 3. So it loses one phosphate. The lost phosphate atom becomes attached to a molecule of ADP, so the ADP becomes phosphorylated to ATP. So now this extra phosphate comes from one of the phosphates of the 1,3-bisphosphoglycerate. And again, remember, this happens twice for every glucose. So 2 ATP molecules are synthesized at this step per glucose broken down. In step 8, this one right here, the 3-phosphoglycerate is converted to 2-phosphoglycerate. So all that happens is that the phosphate moves from carbon 3 to carbon 2. So it's called 3-PGA going to 2-PGA. In step 9, right here, the 2-phosphoglycerate is converted to a molecule called phosphoenol pyruvate, abbreviated as PEP. So the 2-PGA, or 2-phosphoglycerate, is converted to phosphoenol pyruvate, abbreviated as PEP, or PEP. Now PEP is a very reactive molecule, and it's reactive enough to donate a phosphate to ADP. So in step 10, the phosphoenol pyruvate is converted to pyruvate. So it goes from the phosphoenol pyruvate to the pyruvate, resulting in the end product of glycolysis. Now the phosphate, as I mentioned, the phosphate that is lost by the phosphoenol pyruvate becomes attached to ADP, similar to what happens in reaction 7 above, forming ATP in the process. Again, 2 ATP molecules are synthesized at this step for every molecule of glucose broken down. Because remember, there are 2 of the glyceraldehyde 3-phosphates that enter into the reaction at the top. So if we do an accounting of the results of phase 1 and phase 2, I showed that 2 ATPs are invested per molecule of glucose during phase 1. And also that during phase 2, 2 NADHs are produced per molecule of glucose, and 4 ATPs are produced per molecule of glucose. So adding all this up, then the results show that during the breakdown of 1 molecule of glucose to 2 molecules of pyruvate, a net of 2 NADHs and 2 ATPs are synthesized. So there are some important points to remember, or to at least be aware of, about glycolysis. First, each of those 10 steps is catalyzed by a different enzyme, and several of those enzymes are highly regulated, but I'll cover those details in the lecture titled Glycolysis Part 2. Also, all of the carbon is conserved. Glycolysis starts with 6 carbons, all of them in glucose, and ends with 6 carbons, 3 in one of the pyruvates and 3 in the other pyruvate. 3, no oxygen is consumed. And lastly, that there is a downhill trend in the amount of energy contained in the bonds of the molecules at each step. So this downhill trend in the amount of energy allows the reactions to progress. Now there are 3 steps that show large drops in the amount of energy available per mole. And these 3 steps are essentially energetically irreversible. Those 3 steps, 1, 3, and 10, which show a large decrease in energy during glycolysis, are kind of like a river going downhill with occasional waterfalls. And here's a little waterfall at step 1, little waterfall at step 3, and a little waterfall at step 10 in this downhill trend in energy. And so because it's a downhill trend, the series of reactions can occur. In addition, for glycolysis to function or to continue, there needs to be a continual input of the following. There needs to be glucose available. So if the cell runs out of glucose, here at the top, glycolysis stops. There needs to be ADP. And as you see, there is ADP at step 7 and at step 10 required. Otherwise, if we don't have ADP, then this reaction can't continue, or these reactions can't continue. Glycolysis also requires ATP, and you'll notice here at reaction 1, ATP is required, and at reaction 3, ATP is required. Inorganic phosphate is required here at step 6. If there's no inorganic phosphate, glycolysis cannot continue. And lastly, the oxidized form of NAD, which is called NAD+, must be present in order for glycolysis to continue. And you'll see, remember, at step number 6, NAD+ is consumed here. But it turns out that NAD is absolutely critical and can represent a very limiting factor if not replenished. Usually all the other molecules that are required, they're usually present because it's cycled. They're cycled, like the ATP is used, and then that regenerates ADP. And when ADP is synthesized, that liberates inorganic phosphate, and so on. But the NAD is very, very critical and can represent a limiting factor if not replenished. Now, how the NAD is replenished will be described in the next video titled Glycolysis Part 2, and in other videos where I cover aerobic respiration, the citric acid cycle, oxidative phosphorylation, and fermentation. Lastly, I should remind you, as I said earlier, that the end products of glycolysis, those 2 pyruvate molecules down here, those 2 pyruvates, they just can't accumulate forever. The pyruvate has to be removed or processed by some mechanism. And it turns out that this mechanism, or these mechanisms, these potential mechanisms shown by this red arrow with a question mark, are involved in the regeneration of the NAD+ required by glycolysis. And how it happens and under what conditions will all be covered in my next few lectures. So this represents the end of my lecture on Glycolysis Part 1. In Glycolysis Part 2, I will cover details about the molecular structure, the biochemistry, and the regulation of this major metabolic pathway. So thank you for watching and studying with me. And be sure to visit my Patreon website, patreon.com/biology. I look forward to hearing from you.